Good evening, everybody. Just if someone can confirm that you are able to hear me and you are able to see my screen.
Yes, I can.
Yeah, thank you so much. So let's begin for today's session. So today we are going to discuss on transcriptome analysis. What is transcriptome analysis? How it is beneficial for us? And what are the ways why we are doing transcriptome analysis? What are the tools we have to use for transcriptome analysis? So doing why we are doing this? So let us try to understand, because before we do any analysis we have to understand the significance. So the knowledge from the transcriptome analysis we can apply to a broad range of scientific questions, such as gene expression profiling between the samples, identification of allele-specific expressions, disease-associated SNPs, gene fusions to understand disease and their causal variants in different type of cancers and different type of disease. So let us understand one by one what they are. What does they mean? So gene expression profiling between the samples. We can use this information in two ways. Say suppose we have one sample, or say suppose we have one organism. It could be a plant, which could be a human, which could be a pathogen, or anything else. As we know that all these entities are made up of different organs, tissues. So if we would like, and in all these organs, all these tissues, they are different in some aspects, and in some aspects they are similar. So if we would like to understand what is the difference, why they are different from each other, why they are similar to each other, because all these tissues, all these organs, they perform different set of functions. To do those functions, to perform those functions, there is a requirement of different sets of genes. So some genes are present here, some genes are present here, which are unique to organ specific or tissue specific. Some might present over here, some might not present over here. It means that they are only specific to this particular organ or tissue, and vice versa. So this is one way, one application of transcriptome analysis why we are studying. We are trying to understand the gene expression in different organs or tissues. Second questions we can answer. Say suppose we have one disease. We have one normal. So this is person 1 for disease X. We have another person P2 with the same disease. Now we would like to understand what is the difference in this gene expression between two individuals with same disease. Or we have normal condition, we have disease 1, we have disease 2, we have disease 3, or we can say we have treatment 1, we have treatment 2, we have treatment 3. And we would like to understand the differences between all such conditions. So using transcriptome analysis, we can answer all these questions. Now second, identification of allele-specific expressions. Now say suppose I have allele 1, I have allele 2. So I have one kind of mutation I have another type of mutation, or I have one mutation in one copy only, I have mutation in both the copies, or maybe the mutation is homozygous or heterozygous and the wild type. So whatever in the previous section I mentioned, the different kind of gene expression, they could be associated with this kind of scenarios. So if we have the genomic information from the same person or same individual, we can answer this. This particular set of mutation is guiding for that gene expression. So we can analyze vice versa. Third is also similar to that. Say suppose I have one disease. Maybe for this particular disease only single SNP is associated, or maybe over the period of time for the same disease, I find that the another SNP is also associated with this disease. So if we know the RNA expression, we can do the variant analysis from these samples. So we have transcriptomic data. We can call the variant. So this particular SNP analysis is possible when the reference is available. In case of reference is not available, we can do, but we can just do the comparison between the sample, not with the reference. So to do this SNP analysis, we should know what is our SNP in the reference. Then we can answer this question. So in this case, say suppose my reference is like this, and I have one gene expression mRNA read like this. So I know this sequence, what it is. When I will align to my reference, I know in my reference what is the sequence, nucleotide sequence. And if I see this particular gene is highly expressed, and I see the variation between reference, that these are the variations. So then I can directly correlate that, okay, this particular variation is directly correlated with the high expression of my gene. And considering that in the normal sample, this is normal. So they are not mutated. The base change is not there. Gene fusion. So we know that in the cancer, whether it is a solid tumor or whether it is a leukemia, in most of the time it is we are doing this association in leukemias. So we know that all AML, CML, there are fusion transcript. So let us see what is fusion transcript. Say suppose this is our chromosomes. So this is, say suppose 9th chromosome, this is 22 chromosomes. What will happen? Some part will go here and some part will go here. So this particular translocation is called as 922 translocation. So in case of AML, there are several things. So 15, 17 translocations. So a piece of one chromosome will go to another and another chromosome will go here. So such kind of translocation is called a balanced translocation. It can be balanced, it can be unbalanced. So if it is unbalanced, what will happen? The amplification, so gene duplication will be there, copy number variation, we say that. So then amplification of one gene will be there. The classic example is EGFR, which is associated with lung carcinoma. So like that, we have to see all these things. Another classical example is HER2/neu for breast cancer. So these are the classical example of amplification. These are the classification of translocation. So this is we using the transcriptome analysis technique or sequencing. We call it as RNA-seq also. So using this particular technology, we can answer all these questions. So before going into deep, let us see So let us understand some basics of RNA-seq. So what is RNA? RNA is nothing but it is a ribonucleic acid. So it is a ribonucleic acid which is present in all the living cells. It acts as a messenger, means that it carries the information from DNA to the cells to code for the protein which performs the functionality of a cell of a human being or in any of the organism. In some RNA viruses, they carry the genetic information as well, like HCV, HIV, and the newly developed COVID virus. So these are the examples of RNA viruses. In such viruses, they carry the genetic information like DNA from one progeny to another progeny. So basically, the RNA is a single-stranded molecule which which has phosphate, sugars. So what is the basic difference from from DNA? That the base is uracil, which is thymine in case of DNA. This is this organization in terms of the bases and nucleotides. So I'm not going very deep into that because these are very basics and most of us we know. But why I am touching upon it? So before going into deep sequencing of RNA and analysis, we should know some basic aspects of RNA. That's why I'm touching upon few basic points over here. So as we know that from DNA, so let us see what it is now. So we know that, sorry. So we know that in our DNA, which is double-stranded molecule. So what will happen from DNA? It will go into transcription. So transcription is nothing than the generation of messenger RNA, and then from transcription to protein, which is the process is called translation. Now let us see during this process how many types of RNAs are produced. Because for library preparation, for data generation, it is important to know what different kind of analysis from this transcriptome data we can do, and to do so, what are different steps we have to modify at different steps of library preparation, means data generation, pre-processing and downstream analysis. So there are different type of RNA which are very important to know before we go into deep. So there are basically two types of RNA: messenger RNA, non-coding RNA. So means we can divide like this also, coding and non-coding RNA. So the messenger RNA is the one which codes for protein. So it comes as a coding RNA. So this is known as coding RNA because this is the only type of RNA which codes for protein. Rest other type of RNA comes in the category of non-coding RNA. This can be further divided into short, medium and long non-coding RNA depending on the length of the RNA. So short non-coding microRNA, piRNA, siRNA and these all other type of RNAs are short non-coding RNAs. So as we know now, the analysis of microRNA is also important because they are the regulators. So they regulates the coding of messenger RNA. So this is also important to study. Now the another medium non-coding transfer RNA, as we know, it performs the action when translation happens. SNO-RNA, snRNA, single short coding non-coding RNA. In the long non-coding RNA, there are various forms of depending on their production, their action, their structure, and what kind of influence they do. So like intergenic, intronic, sense, antisense. cis RNA, trans, so all that, and linear, circular based on their shape and influence. They are non-infectious or infectious, which kind of elements they have, replicative, non-replicative, dependent, independent, and dependent, like that. These are the several categories, and whatever is our intention to study, based on that, we can choose the downstream process. So we will see how, if we have the intention to analyze all these kind of RNA or some specific kind of RNA, how to choose the downstream process to proceed further. So why do we need RNA sequence analysis? So as I mentioned before in my first slide, that there are several questions. If we do the RNA sequence analysis, we could answer. Now from the technical point of view, for other point of view, there are some more things which we need to understand. So if we say that why do we do the RNA sequence analysis? So if we know we have DNA, so what we can answer from this DNA? We can see the variation in the genome. But how it is influencing our proteins or the RNA, we cannot answer directly. But how we can correlate this variation using the RNA-seq data. So as we know, say suppose this is our DNA. We know that our gene can be coded in 6 frames. So it can code from here, 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 if it can code from here, or it can code from here. If we have the DNA information, we won't be knowing which frame it is coded for. But if we have the RNA sequencing data with us, we can go back and infer that which frame this DNA will code for our proteins. So this is the prime advantage of doing the RNA sequencing analysis. And directly, if any variation we found here, we can directly go and check in our DNA for that particular variation only, whether it is coming from DNA or it is coming during the process of transcription or post-transcriptional modifications or post-translational modifications. So doing so, we can go back and check. But if we start from here, it is a bit difficult to infer. So this is the advantage of RNA-seq. We can move in the forward direction towards the protein, and we can go back and infer the information of DNA. So the RNA sequencing is sensitive. It is very precise and accurate tool for measuring abundance and gene expression across transcriptomes, which I explained already how between different treatments. Say suppose I have one species or I have one tissue or something X, I have some X. I would like to increase the probability of Y in this set. So what I am going to do? I can choose some 5-6 treatment which can enhance the expression of Y. So for the same X, I can treat with the T1, I can treat with T2, I can treat with T3 and so on. And I can see that how they are different. So I can choose which treatment is working for this to enhance the factor X. So which is not possible on DNA sequencing. This is possible. This is possible to study only via RNA sequencing. So characterization and quantification of the genome. As I explained, this is RNA. If we know this, we can infer the information of protein and we can infer the information for DNA. Genome-wide analysis of transcription at single nucleotide resolution. This is again same. With DNA, I won't be knowing what the genes and how they are expressing and how they are affecting the proteins. But if I have, I can interpret my proteins, I can predict the structure also, and I can predict my genome as well. And as we know, considering the third point, alternative splicing. So what will happen? Say suppose this is my gene. It will have several exomes, several introns in it. So what will happen? If I know this RNA, say suppose in my normal condition, this is my usual transcripts. In my normal condition. But say suppose if any mutation is happening or something is happening during the process, my transcripts will be like this. So this is called alternative splicing. So this transcript will not form. Instead of that, these transcripts will form. So doing this, we will know that, okay, this particular mutation is happening, so that this splicing is happening, and this is related to my disease. So I can provide a treatment so that I can try to repair this particular phenomenon, so that alternative splicing will not happen, and it will return back to the normal splicing. Which is, this phenomena is not at all possible to study from DNA data, but RNA, it is very much possible. So post-transcriptional RNA editing events. What does this mean? Say suppose my DNA is normal, but I see that my protein is not normal. So now where I have to look? My genome is normal, I cannot do anything. So there are post-transcriptional modification and post-translational modification. These are the two important steps where the variations also might come. So what will happen? My messenger, my RNA is normal when I study this. When this is normal, my protein is abnormal. What does this mean? This is normal means this is normal, my gene is normal, but still my protein is affected. So it means that there are some modifications are happening after transcription and during the post-translational modification, so that my protein is altering. Gene fusion already I have explained, SNPs already I have explained, and allele specific already I have explained. So what does this mean? RNA-seq investigations can be applied to different populations of RNA as different type like mRNA, tRNA, ribosomal RNAs, right? So microRNA and so on. So now the another important parameter in RNA sequencing, there are two steps in RNA sequencing. One is the data generation. And second step is the analysis. So data generation involves experiment, samples. It starts from sample, then experiments. Experiments involves library preparation, different type of library preparation, and the sequencing. So this is my data generation. Then comes to the analysis. So this sequencing is generated in the form of intensity, which directly comes out from the machine. So before proceeding for the analysis, what we do the base calling, and after base calling, so these intensities are converted into ATGC, sequence of ATGC using the base calling script. And once we have this is stored in terms of FASTQ files. And from this, we go for the different kind of data analysis. So let us see all these steps one by one. So let us see first the experimental design. So in experimental design, there are few steps which are very critical. So first comes the RNA. So from where our RNA is coming? So RNA source is important, because if it is coming from blood, it will be very good. But again, how much time do you collect and what is the time gap between the collection and the RNA extraction? If it is fresh tissue, if it is old tissue, if the tissue is stored in some RNA later, or if it is extracted from FFP tissue, and along with the source, what method do you choose for the isolation of RNA? So our RNA quantity and quality depends on all these factors. So our RNA should be having high ring number. What is ring number? Ring is nothing but the integration number. Integration means how intact our RNA is, whether it is fragmented or whether it is intact. So now second is the depletion of ribosomal RNA. We know that when we isolate the total RNA. So this is our total RNA, which will have our mRNA, which will have our tRNA, and which will have our ribosomal RNA. And we know that the maximum proportion occupies by ribosomal RNA. So we have to deplete this. Usually tRNA will not affect in the analysis and sequencing, but so we have to decide the method when we are preparing the library, which method we have to choose, whether we are enriching our mRNA or depleting the ribosomal RNA. So we will see what to choose, what not to choose. Then the library is specific. Which strand? Are we preparing the library strand specific? Whether it is 5' or 3' or 3' to 5'. This is very specific. If we have the gene information, which strand it codes for, we can decide on strand specific. But if we do not know, then we can prepare blindly and then use it. DUTP method. Sequencing depends on the analysis goal. Again, what is our objective? The sequencing depends on that. Whether we are going for paired end, whether we are going for single end, whether we are going for 75 base pair, 50 base pair, 150 base pair. So that all depends what is our objective to do. We will see these factors in later slides. We will discuss all these factors in detail in subsequent slides, because this is very important before we proceed for the RNA-seq data generation and analysis. Then comes to sequencing depth, how much data we have to generate in terms of read numbers. So medium to highly expressed gene, 5 million reads. If we know that we are looking for some genes which are not highly expressed or which are very lowly expressed, then 100 million reads are required. In single cell highly expressed genes, 1 million reads, 50,000 to 1 million reads are enough. In differentiating cell types, 20,000 reads we have to generate. So now let us see. There is important factor replicates over here. So why do we need replicate? Let us understand what is the importance of these replicates. So say suppose I have one tissue X. Then what is my aim? We are doing the RNA extraction. First thing. the data generation depends on the amount of X, how much tissue I am taking. Then how much RNA I am getting from this. Then what is my gene expression in this sample? We do not know anything. And then we are sequencing it. So sometimes our sequencing is very good, sometimes it is not good. One time today we are sequencing something else, it will come. Tomorrow we are doing something else might come. So to avoid all these errors, we need to make the replicates. So ideally 3 replicates are required. So these replicates means 3 different individuals or representative of this tissue. So take this tissue. So again, if we take the same tissue from 3 human beings, they will be entirely different. That is the biological system, biological variation. So we cannot avoid this biological variation. So what we do? We take one sample, we divide into 3 portions, then we called it as replicates. Now another way, I have one sample, I have extracted RNA from this, and then I can divide this RNA into different portions and prepare the library. So ideally, we have to go for this kind of replicates to avoid all these errors. And when we have replicates, we have to put in different lanes of the instrument. So this is our flow cell, because to avoid the lane error. So one sample we can sequence here, one replicate we can sequence here, and we can pull the data. We can take the mean of it. Say suppose I have one tissue, say suppose treatment 1. For this treatment 1, I will have a replicate 1, replicate 2, replicate 3. I have another treatment, say, and then for that also I will have replicate 1, replicate 2, replicate 3. And when I am doing the analysis, I have to pull this and then take the mean of it. So this is the meaning of our replicates. Now spikes in. Say suppose my gene, I want to see the differential expression of genes. I got some expression over here. Say suppose I got some expression here, some expression over here. Now how I know that this is a real expression? I do not know. What I want to see, yes, what I predicted, it is coming here, and what I predicted over here, it is coming here. But how do I validate that? So that's why we do spikes in. So what is spike in? There is a sample. We know after RNA, so this is already known RNA, and we know that in this particular sample, the gene expression values will be this. If we take such X amount of RNA, the gene expression will be like this, A, and for the another gene, the expression will be B. So when we compare this, when we spike in this, so the same amount of expression I should get here. So if I am getting the same expression in the samples also, it means that my experiment or my sequencing data is good. Randomization of library preparation and sequencing run, which I have already explained that how you can randomize your library preparation. So treatment 1, take 3 replication, treatment 2, take replication. So if you are taking, so say suppose I have one cell, I am plating in 3 plates. So this is my 3 treatments. So this is a replication of this. So this is a biological replication. If I cannot take this, so this is important. One should not avoid this. But when we are doing the method validation or method development, along with this, we have to take a technical replicates also to avoid the method errors. Say suppose I am plating my cells in 3 plates. So this is my treatment 1, treatment 2 and treatment 3. So now for my method also I have to divide into 3. Everything I have to divide into 3. So A1 T1, A1 T2, A1 T3. So this also I have to divide 3, I have to divide 3. So this is my technical replicates, this is my biological replicates. So while performing an RNA experiment, we have to take care of these biases. So now, in terms of the data, we have to do the quality control. So we have to see look at our data, which is raw reads, read alignment, quantification, and reproducibility. In the next point, let us see one by one all these steps. Source, as I mentioned before, the source will also give the variability for your RNA. Say suppose you have FFPE for the same kind of tissue from different sources, it will give you variability. If you have kidney sample, if you have liver sample, if you have gut sample, or any kind of tissue sample, any kind of organism sample from where you are getting your RNA. In each type of tissue, in each cell type, the amount of RNA will be different. So one thing is the RNA amount will be different, and say suppose you are taking the cells at when they are just newly formed, they are maturing and they are getting older. So then in these 3 type of cells also, the RNA amount will be different. So one should take care of such instances. Starting material, how much amount of tissue you are taking to get the appropriate amount of RNA. Preservatives used, how you are preserving, whether you are preserving in saline or you are preserving with snap freezing, you are storing in liquid nitrogen, you are storing in RNA later. Again, there are different type of RNA laters. So how you are choosing your preservatives and what is your storage? Say suppose you have sample 1. You are extracting RNA after storing of 3 months. You are extracting from the same sample after 6 months or 1 year. So you will get a different kind of yield from the same source after different time points. So when you are starting for the extraction, please take care of all these steps. Method of use. What the method you are choosing for isolation. Whether you are using triazole, whether you are using the conventional phenol chloroform method, whether you are using the kit. So you need to see which method you are using, because if you are using triazole and if you are not separating your layers well, then some contamination will come. If you are using phenol chloroform, then phenol contamination might come, protein contamination might come. If you are using kit method, so you need to see which kit you are using, whether for FFP you are using the appropriate kit or not, you are removing the wax from it properly or not. So all these factors plays an important role to provide the quality and quantity of our RNA. So once now we have RNA, so this RNA, how to check the quality and the quantity? So there are usually 3 methods. One is the spectrophotometric, colorimetric, and the capillary based. So what we are doing in this spectrophotometry, we are measuring the quantification based on the absorption. We are measuring the 260 by 280 ratio. We are measuring the 260 by 230 ratio. We are measuring the other contaminants in our RNA. So we are measuring whether protein contamination is high or not. We are measuring whether DNA is there or not. We can run the gel also and see if any contamination is coming from DNA or not. So say suppose this is my RNA, DNA will always be here when we run on the gel. Fluorometric quantification, what does this mean? It means that again, this is using some fluorescent dyes, we are seeing that we are measuring exact quantification of our intact RNA. So RNA integrity by bioanalyzer. So this is a capillary based. When we run on the capillary, then we can see the specific two bands, 23S and 18S. So usually this is for eukaryotes. You can see two bands in the capillary. So if these two bands you can see, it means that your RNA is intact and proper. If not, then it means that it is degraded. Again, molecular weight is one parameter for RNA integrity. Here you can quantify also and you can see the integrity of your RNA. So now let us see what are the different factors which affects the transcriptome analysis. So as for the experiment also for isolation of RNA, as there are multiple factors, for analysis also there are multiple factors. Before we go into that, we have to see all these factors. We are not audible. Now I am audible.
Yes, ma'am.
Yeah, okay, fine. So for the analysis also we have to choose different factors. What are the different factors? So again, data analysis again depends on several factors. RNA quantification. Say suppose I know that my genes are low expressed. To see these genes, I have to take the appropriate amount of RNA. If I am taking 1 microgram of RNA, then I am able to see that my genes are coming. My RNA expression or lowly expressed genes are coming. But if I am taking only 1 nanogram or 100 nanograms, how my genes expressed genes will come in my data. So there are two ways to do. Either take a more amount of sample and do that, or if the starting material is a constraint, then we have to generate the more amount of data so that we will be able to see our lowly expressed genes. Transcript identification. So this goes with both the things, RNA quantification and identification. Reproducibility. What does this mean? Today I have sequenced one sample. I got some genes which are expressed. The same sample I have sequenced another time. So I should be able to get the same profile. If same sample I sequence third time, I should be able to get the same profile. If I am getting the same kind of profile, then it means that my analysis or my experiments are reproducible. Otherwise, no. Alignment. If I am doing reference-based alignment, then I have to check my alignment, whether my alignment is perfect or not. Say suppose this particular gene is located here. So my transcript also go and should align here, not here. So this is, we have to check during the alignment. De novo transcript construction. What does this mean? It means that we do not have reference available. And when we prepare the assembly from our reads and do the annotation of this should give the transcript reconstruction. So when we first align our reads and now we have to consider this as our reference, and when we remap our reads back to this alignment, So they should go and alignment, give the good alignment here. So that is the de novo transcript reconstruction. Differential gene expression, same. If I am able to see the differential expression in one time, another time also when we sequence, the same profile we should get, and so on. Alternative splicing. So say suppose in my sample first time I am able to see alternative splicing. Many a times I sequence, we need to reconfirm that. Then the visualization, gene fusion, small RNA, functional profiling with RNA-seq. What does this mean? Functional means if we do the ChIP-seq or methylation analysis, we will come to know and compare with the RNA-seq. So what will happen? So say suppose I do the ChIP-seq analysis. Let me clear this my screen. Okay. So what will happen? So what is ChIP-seq or methylation? It is a DNA protein interaction. Right. So we know when we do this analysis, we know that where this protein and the DNA binds together. And I know that if my RNA is highly expressed or low expressed, then this I will get to know. Integration with other data type, this is the other data type. So I can, this RNA expression, I can integrate with my methylation data. I can integrate with my ChIP-seq data. I can integrate with the single cell data. I can integrate with the metabolomic data. And on the other hand, I can integrate this with my DNA or genomic data as well. So this is all I have mentioned: DNA sequencing, DNA methylation, chromatin, microRNA, proteomics, metabolomics, single cell, long read, everything I can integrate with my transcriptomic data. So now let us see what kind of different type of libraries are present and how to take care of those, what to choose, what not to choose. So there are two types of libraries, directional and undirectional. So if we make the directional library, so we know that we are preparing from 5 prime to 3 prime, or we are preparing from this direction. So we are preparing in this direction or we are preparing in this direction. We know in advance. So say suppose I am preparing my libraries in this direction. But if I get the alignment in this direction, it means that this is a contamination because I didn't prepare anything in this direction. So like that. And if I know the direction, in which direction I have prepared my libraries, I can inference that from which strand my genes are coded. Where is my gene located? But this might be very challenging or may not be possible for the low input of RNA. So when you have the high amount of RNA, better to go with directional. If you have low input or sample constraint, then don't go for directional, go for undirectional library preparation. Messenger RNA versus total or depletion. Now what does this mean? So as I mentioned to you, we have coding and non-coding. So this is our coding, this is our non-coding. Except mRNA, everything comes over here. So if we want to know the population of different kind of RNA, then don't go for enrichment, because when you perform these different steps, automatically our messenger RNA will go away and we will have the another pool of RNA. If our interest in other type of RNA. But if we want to study along with messenger RNA, then we have to go for total RNA sequencing. But if our interest is not at all having the study of non-coding RNA like transfer RNA or ribosomal RNA or long RNAs, microRNA, then better to go for mRNA enrichment or rRNA depletion. Already we know that, so there are two common approaches which people use, RNA depletion or mRNA enrichment. If our intention is to study the mRNA, better to go for mRNA enrichment. If we want other types of RNA to be studied, go for rRNA depletion. So if we are doing the enrichment, how we are doing the enrichment? We are using a poly A tail. But if we are doing poly A tail enrichment, so link RNA will not come. You will lose your link RNA because they don't have the poly A tail. So active transcription versus standing mRNA pool. So this we can get from the mRNA enrichment. So one should take care of these factors, how we will then only decide which kind of libraries we have to prepare. There are different protocols. So now what type of sequencing we have to go for? So again, this depends on the interest, the expression, quantification of known genes. If we know, our intention is to know only quantification of known genes, so 50 base pair single end is absolutely perfectly fine. We don't need to go for 75 base pair or paired end, long read, because that will give you a high cost. So if we have a cost constraint and if the interest is just we know what are the genes are expressed in this particular disease state and we would like to see in different biological states the expression of the same genes or in different treatment condition or disease states, we can go for 50 base pair single and read library preparation, sequencing data generation, and that would be absolutely fine. But if we would like to see the splicing, alternative splicing, then 100 base pair single end is also fine. But longer read, go for longer read if you want to see for the splicing. Because sometimes what will happen, we are doing 100 base pair. But even if our splicing is happening somewhere here, we might miss. So better to go for little longer base pair read. Again, if our splicing is site is present between the repetitive regions, then again we will miss that with this short read. So better if we know the splice site, which kind of a splice site we are looking for, then decide whether you want to go for short read or long read. So if we want to do for some novel transcript or per transcript expression or some novel variants, then 100 base pair, paired and read is a better choice to go for. So now let us see how many reads are required. So this is the length. Now how many reads are required for doing the proper data analysis? Typically we aim to 20 million reads for human and mouse size genome. So as we know that these organisms have 3.2 GB genome, human, and approximately the same for mouse. So if you have a genome size of this is the optimal number of reads we have to choose. If we are doing some de novo discovery, we would like to see some novel transcripts, then 50 to 70 million reads or more. Even in case of lowly expressed transcript, this is the optimal number of data we have to generate. More replicates, more useful, then more reads. So what will happen? Say suppose we have only 2 replicates. So we will have a data from 2 replicates. But if we have many replicates, so there are 2 ways. You take only one sample, generate more data, not of any use. You take more replicates, generate little less data, more useful, because ultimately you have to pull this when you are doing the quantification and then use it. So this is more insightful rather than this. So replicates. As I mentioned before, we have to choose the replicates widely. So compared to array, RNA-seq is a very clean technical measure of expression. Generally, do not run the technical replicates for RNA sequencing data, but one must run the biological replicate. For clean system like a cell lines, optimal are 3 into 3 or 4 by 4 is very common. Higher number is required when the system is more variable. Always plan for at least one sample to fail. So say suppose if you are choosing only 2 replicates and one is failed, you have only left over with 1. So if you choose, so we are aiming minimum is 3 by 3. So if we choose 4 and we expect 1 to be failed, at least we are left over with 3. So choose wisely. Randomize across sample groups. So you have to choose randomly. It is not like that you are choosing deliberately. You have to choose randomly. So the examples, so as I mentioned to you, cell lines are very clean. They are very easy. So you plate some maybe 1 million cell in 3 plates and take it, pull it, or whatever the way you choose, because the variability will be less. But when we come to the primary cells or a biological system where the system is more variable, so we have to take the more random sampling when the variability is high. So now comes to the library preparation. What do we need for the library preparation and how much amount of input RNA is required? So here I am presenting with one table which gives you a glimpse of different kind of library preparation kits are available, and for each kit what is the input amount of RNA can be used. So this is the workflow and how what is the amount of time it requires to prepare the library, and which kind of analysis you can perform using these kits with the input of this much of amount of input RNA. So now let us see what are the different principal components in library preparation. So first is our of course the RNA. Then what is the minimum quality you will get? So if you are doing the true-seq stranded one, complete RNA, seq V2 version 2 or smart. So 1 nanogram depleted RNA, 0.5, 100 nanogram and 100 nanogram depleted RNA. So what is the minimum quality from different samples? What we are looking for, because FFP we already know it will be degraded. We don't look much into the integrity of RNA, but for other sample, RIN number should be more than 7 is preferable. 8 is very good, but so because FFP you might get 2, 3. So we have to work. There is no other choice. RNA depletion. So with these particular kits, we require RNA depletion. With this, we don't need. cDNA synthesis, how, on what principle? So it uses random primers, selective primers. It can choose random or oligo DT and random primers. Fragmentation. So we have, so once we have RNA, at the first step, we don't fragment it because this is very unstable and already it will be little fragmented. And from even after converting the cDNA from the FFP sample, we don't fragment because we already know that it is fragmented. So when we have RNA, first we convert into cDNA and then fragment it because cDNA will be very stable. So I advise if somebody is doing RNA sequencing data analysis or preparing for the data generation, don't do the fragmentation at first step. First convert into cDNA and then do the fragmentation. Strand selection, library preparation methods, multiplexing, and the experiment duration. So this multiplexing is also very important. As we know, This is the first advantage of NGS. We can pull different samples from different biological systems using a technique called the adapter ligation. So each sample will be ligated with specific adapters with the known sequence, and then we can separate them after sequencing data generation. So let us just quickly look at what is how we do the library preparation. So the library preparation, as I mentioned, we have total RNA. We can choose two methods: mRNA enrichment using poly A tail or rRNA depletion. So it is your choice whether you do the RNA fragmentation or not here, but usually we don't advise. First we prepare the cDNA and then do the fragmentation, then the adapter ligation, and then doing the clonal amplification, and then doing the sequencing further. So amplifying the fragments like this. So we have RNA, we have cDNA, we do the adapter ligation, and then do the clonal amplification of this, and then doing the sequencing. So now quality control. So these are the different kits for this is the quality control for the library sequencing libraries, because every step we have to be sure before proceeding to next step, to ensure that yes, whatever we have performed in that particular step, it is up to the mark and ready to go for the next step. So this is for the library. Once we have prepared our libraries in as per the previous slide, then again those libraries need to be passed through the quality control. So there are different methods: fluorometric method, real-time based quantification, and insert size by bioanalyzer. So fluorometric, you just do the quantification. Here you cannot measure your size. Real-time PCR, again it is a quantification. You cannot expect the size. Bioanalyzer, as you are running on the capillary, you can measure the exact size of your library. So what you expect, you know that what is your fragment size, how much fragment you have uh, generated. So that much fragment size you will be able to see that peak here. So here you can even see whether in your library adapters are present or not, because before final library preparation there is a step you can remove these adapters. So which is time dependent cleaning procedure. So you optimizing those steps, we can remove the adapters from our library, because if they come in the sequencing data, we cannot ignore them while analysis. We have to remove. If still during the optimization of library sequencing, we are unable to remove these adapters because of some reason and it comes into the sequencing data, there we have to definitely remove before we proceed for the downstream analysis. So now let us see what are the factors influence the library preparation. So again, there are different factors like RNA species, as mentioned, mRNA, rRNA. So if we are looking for mRNA, but in our library ribosomal RNAs are more, then it affects the analysis. Selection of small RNA species, complementary DNA synthesis, multiplexing. So say suppose I am pooling 4 samples. I am taking this sample as 10 nanogram, and this I am taking 50 nanogram, this I am taking 25 nanograms, and this I am pooling as 100 nanograms. So definitely when I will do the sequencing, because I am pooling these samples, definitely this will come as more and this will be coming as less number of reads. So we have to pool in such a way that all the samples will be same and then gives the same kind of data generation, equal data generation. Quantitative standard, as I mentioned to you, spikes in selection of tissue or cell population and the tissue heterogeneity. So those things also will affect our library preparation. So now coming to the data generation and analysis. So there are different kind of Data generation already we have spoke about, and now let us go for the analysis. What different kind of analysis we can perform on whatever the data we have generated. So for the short read, so there are two kind of data usually we generate from the sequencing. It can be a long read or it can be a short read. Short read, we can do alignment, we can do transcriptome reconstruction, we can do de novo transcriptome assembly, alignment free quantification, differential expression analysis, fusion prediction, variant calling, RNA editing prediction. So these are the kind of analysis which are very common from short read data. Long read data, after error correction, we can do alignment, we can do transcriptome reconstruction and fusion prediction. Right now, we have not validated for all variant calling and other things, but it can be done. So we need once if somebody would like to do other kind of analysis on long read, then they need to validate those tools and use them. But it is not impossible, it is possible. So there is a common general workflow from data, what the data we get, irrespective of whatever kind of data it is. So first is the genome mapping, then transcriptome mapping or the reference-free assembly. So this goes when we have the reference available. This is when we do not have the reference. Reference is not there. So what does this mean? So when we have transcriptome, we are mapping to the genome. We are not mapping to the transcriptome. We are mapping to the genome. So what we have to do? We have RNA-seq data. First it will reverse it and then it will map to the genome. So reads. So there are two common pipelines available. Top hat, star, mapping to genome, cufflinks for the quantification, and so on, and then the functional annotation. If we are mapping to the transcriptome, then we have reads, because we are mapping only to the transcriptome, so we can use different kind of aligners depending on the read length, and then map to the transcriptome and just quantify the transcripts. If we do not have the reference with us, we have to do the assembly first. So we are having the reads using a Trinity pipeline. We build the assembly of the transcriptomes. Then whatever the reads we are having, we are mapping them back to this assembly using the aligner bow tie and then calling for the transcriptome quantification and then the functional annotation. So this is a general workflow. I am not giving very insight into this, how we use, what we have to do exactly, that we will see in the subsequent steps. So, but this is the overall general workflow which can be divided into these three kind of analysis. So data analysis, as I mentioned, there are when we have the reference, when we do not have the reference. So what are the factors, different factors which affect my analysis? Quantification of short transcript and low expressed transcript. We have to be very, very careful when we report these low expressed transcripts. Impact on gene structure on the accuracy of quantification. Say suppose I am accurately quantify of one gene that it is upregulated. Why it is upregulated, that again reflects in the structure of my genome. So what I see here, that should directly reflect in my gene structure. So we have to be careful about this. Impact of sequencing depth and relative abundance of the quantification. Say suppose I have two genes, one is here, one is here. Say suppose by any chance I am having more depth over here than here. Definitely I will see that this is highly expressed, this is low expressed. By looking at this grossly, I can say, anyone can tell this. So to avoid all these kind of errors, we have to normalize our data before we go and do the exact quantification. So there are different kind of methods by which we can normalize our data. We will see that. So I'll just take a 5 minutes break over here, and any question meanwhile, just please ask, unmute and ask.
Okay.
So you can ask the question, no problem. Any question?
For me, no, thank you.
Okay, so then we can proceed further. Yeah.
Fine by
So,
me.
sorry?
It's completely fine by me.
What about others? Any questions so far? Any doubt? Okay. So if no question, then we can just start further. So now, as I mentioned, we have raw data. Now our data is out from the machine. Now that is what exactly we are looking for our analysis. So these are our reads. Each read will be stored in the form of FASTQ. So what is this FASTQ? Let us see. So the FASTQ file is nothing but it is a 4-line sequence. So in the first line, it starts always with @ symbol and it will have a sequence identifier. So this is unique for each read, each file, each sample. So if it is 1 over here, it means that this is single end. If here it is 2, it means that it is paired end. So this is, so every bit of this sequence identifier has some meaning. Second line, it will present with the sequence. So whatever the read is there corresponding to that sequence, it will be. Third line will start with the sign of plus. This is nothing, this is called the quality header. It might be empty. If something is written, it will be more or less similar to the first line. And the fourth line is nothing but it is your Fred score for corresponding to each base. So this is the structure of our FASTQ file. So this is, we call it as raw data file. So once we do the quality, we will see how we do the quality and other things, what the tools we use. First, let us understand the process, because the understanding of the process is also very important. So in this, so once we have data, we have done the quality, and then if the reference is available, we are mapping to the reference. Now let us see how it looks. So I told you our gene is, if we are mapping to the genome, how it will be. So we have a genome, we have gene. This gene will also have a different segments, which are represented over here as exon. These are the introns. This is exon, these are the intron. So now we have only the transcripts, right? So we have transcript. Our transcript will map only here. They will not map here. So this is how exactly it is showing over here. So if something is mapping between the exon, it is not correct mapping. If it is mapping partially with the exon and partially with the introns, this is not correct. So our tool should know where it should go and map. Say suppose our read is there like this, right? So this part might come from here and this part might come from here. So our tool should know this is the splice junction from where my read will splice. So what is the funda beside this? Till date, all these splice sites are known. Splice sites. So whatever the genes, whatever the transcripts are there, we know where they are having these splice sites and what are the sequence of these splice sites. So this information is stored in our tool. So the tool is known as splice aware aligner. So there is a tool called top hat or star. These are the splice aware aligner. They are nothing. They are nothing. In this there are two components. First, the tool breaks your read wherever the splice sites are there. And then you have an aligner which is bow tie, and this bow tie helps to align these reads to your genome. How this is work? So in first step, your splice aware aligner, this script will break your reads wherever the splice sites are there. And you know now that because we know that at which direction my splice site is there. So this aligner will correct and give the proper orientation to this particular segment or this particular segment or this particular segment, which can go and now align to the genome. Understood? So this is how this reference mapping is worked. Now in another case, as I mentioned, there are another kind of mapping where we are mapping just with the transcriptome. So when I am saying transcriptome, I know that I have only the functional transcripts and I am just mapping my reads over here. So I don't need the splice site aligner. I can just use a simple aligner, that is bow tie or any other aligner which can just align my reads to these transcripts and use it. So if I am just not aligning and just doing the quantification, I can use high set. If I am doing alignment based quantification, I can use either star or top hat. Now I have aligned my reads to my reference. How do I know that my alignment is good or not? To do so, there is a tool called QualiMap. As the name itself indicates, map. So this is doing the quality check of the mapping reads. So how it gives the stats? Left reads means this side read, this side read, how many are pairwise alignment, everything it will give. So input reads, mapped reads, how many reads have multiple alignments, right side lead, how many I have put, how many are mapped, and the overall read mapping. Aligned pair, how many aligned pairs? So this is my pair. How many are pairwise aligned? So if I see that 94% is my concordant pair alignment. So it means that it is very good alignment. If we see only 60%, 70% alignment, that is not good. So again, depending on our objective, we can take a call on that whether my alignment is good or not. But generally it should be more than 70% my alignment, then it is a good alignment. Otherwise, we have to go back and check where is the problem in our alignment and do the realignment. So once we have done the alignment, so this is we have some numbers. How we are sure that this alignment is good or not? So it is a general phenomena. If we visualize something, if we see something, we have more confidence. So to do so, to see our alignment, whether our alignment is good or not, we can visualize the alignment. So this is how our alignment looks. So there is a tool, there are tools, not is tool. So like one very common tool is the IGV tool, where you can visualize your alignment. So you have your reference, you have your aligned file, which will be either in the BAM format or SAM format. So usually SAM takes a long, so more storage. So immediately as soon as we have alignment file, we convert into BAM file. So this is nothing but this is the binary file of SAM file. So this BAM file, with the help of reference and GTF file, we can visualize like this. So say suppose if this is my genome, if my reads are like this, so this is perfect alignment of my genes. If in my data I see one read is coming like this. So it means that this part is deleted. So deletion is there. If we see that our reads are far apart, means like this, it means that there is an insertion over here. So like that we have to see and then visualize our data. So read over exons, reads over introns. If any reads are mapping on the introns, we have to correct them. Reads in the intergenic region or the strand specificity. All these factors we can visualize and get the confidence when we visualize our data using different tools. So quantification, when we would like to do the quantification of our transcript, how we can do. So say suppose this is my exon 1, this is my exon 3 and this is my exon 2. So this is my first splice form, this is my second splice form where my exon 2 is not there. Now I would like to see definitely splice form 1, how it is. Correct. This is my second splice form. And if my reads are there like this, I have ambiguous data. I can't say that whether my splice form 2 is there or not. So we don't have reads over here. So this is definitely, this splice form is there because exon 2 is not there. So this is read. How we are aligning the reads. So now let us look for the different kind of quantification. So simple quantification. Count the reads overlaps with the exon of each gene. So these are my genes. Say suppose this is one gene, this is one gene, this is one gene. So we have to count the reads. So in each gene we will have a different exons. Correct? So we have to count the reads which are overlapped in both the exons or whatever the exons are present. So this is a simple quantification. We just count the reads and see how the quantification is happening. Consider the library directionality. So when we are doing this quantification, we have to see which direction our library we have prepared. If we don't have that information, we can say unknown and then it quantifies, the tool quantifies. So gene level quantification, many programs are there. Seq-monk, when you can graphically see the quantification, how many reads are there per gene per exon using the tool called Seq-monk. Feature counts, bed tools, HT-seq. So using those different tools you can quantify. Analysis of splice sites. So there are different tools by which we can do that. Cufflinks, RSCM, BitSeq. Junctions, counts plus DSeq or HR. Compare the splicing decision ratios. If we want to see the splicing, say suppose I have 2 different or 3 different kind of splicing. So I can, using these logistic regression or RMATs, we can take the decision ratios. Say suppose this is 10%, this is 20%, and remaining is this percent. So we can see what is the percentage of each splice sites using logistic regression. Now, as I mentioned that we have to normalize our data. To do so, there are different terms we use for doing the normalization or calculate or quantify the transcripts. So first is the RPKM. It is nothing but the reads per kilobase transcript per million of library reads. Understand that what is this. We will see the formula also. So this corrects the total library coverage. So in this, it considers both the factors, our read length and the number of reads. So it corrects the gene length. As we know, each gene will have a different kind of length. So it corrects the quantification. So sometimes what will happen, if the shorter genes are there, they will be more. If the longer genes are there, the reads will be less. So based on the read length also it corrects, and based on the total depth of the data. FPKM, fragment per kilobase of transcript per million fragments of the library. Here it won't consider the gene length. It just consider the depth of the data. TPM, transcript per million. This is very simple. It just gives you the quantification based on per million of data. So this is more common because it won't consider, because definitely we know that the gene length is different. So why we have to normalize this? We can't ignore this factor. So it just normalize the data based on the per million reads. So let us see what are the different formulas to do so. So RPKM, number of reads mapped to a genome into 10 to the power 9. This is GB, and total number of mapped read x gene length in base pair, as I mentioned to you. So it corrects with the gene length and the genome size. TPM, just count per million or per million, based on per million, nothing else. Irrespective of your genome size or the gene length, it just normalize the data on the basis of per million reads. FPKM, number of reads mapped into 10 to the power 9, that is the genome size, gene length, and total mapped reads. So these are the formula. If someone would like to calculate, one can use these formulas to calculate. Otherwise, tool gives automatically these FPKM and RPKM values, but we should know how the tool calculates these FPKM, RPKM values. So now, what are the different steps involved grossly in the analysis? So as I mentioned before, there are two pipelines. One is using top hat, that is a reference based, and one is the Trinity pipeline, so which is when the reference is absent. So top hat is a aligner which integrates with your bow tie. So the entire pipeline is known as tuxedo pipeline. And the another, when reference is not there, that this pipeline is known as reference pipeline. So tuxedo pipeline and star pipeline is very common for the RNA data analysis. One can choose anything because at some steps they are common, whether you choose star or tuxedo. So as a first step, as I mentioned, raw data QC, alignment, assembly of the reads, merging, differential expression, annotation, visualization, and then the interpretation. So we have reads from two different groups. We have done the quality separately for both the groups. We have aligned separately to the reference. So group A group B. So we have transcript from here, we have transcripts from here. We need to see using the coupling, we can merge and then see how the genes are present in both the samples separately. So we can merge using couplings, cuff merge. We can do the differential using cuff diff, and then we can visualize using cuff bound. So this is the general pipeline overview of tuxedo pipeline, which is having a top hat aligner. So there are different tools which I have shown here. So we have raw data. Using fast QC, we can do the QC of the raw data. Once we have the QC file with us, we know whether the adapters are there or not. So as I mentioned, if you remember in my previous slides, I have discussed that adapters. So during library preparation, at the last step, using different differential hybridization points, we can remove the adapters. But if we are unable to do so, we can use cut adapt to remove the adapters. So to remove the adapters, we should know the adapter sequence. So this sequence is specific to each kit or the platform. So when you procure the any reagent kits or the adapters for the sequencing, your vendor will provide you the adapter sequence. You know the adapter sequence before in hand. So once you have removed the adapters, then there is one parameter Q. So our read quality should be more than 30 or equal to 30. So as I shown you before that the first line is with at, then our sequence, then plus, and then in the fourth we have the quality score, which is known as Fred score. So there is one table called ASCII. We can match this with the ASCII table, and then this Fred score should be more than 30, equal to or more than 30. Usually everybody recommends, but for RNA-seq it can be worked out with 25 as well. So that depends. So then if we have to filter the reads, so there are combination of tools, cut adapt, NGS QC tool, we can use them. Here I have given you the code and this link also, how you can use the command line using for the terminal, Linux terminal, and you can download these tools and install on Linux. Usually they work with Linux, not with Windows, but fast QC someone can download on the Windows and use it. But cut adapt and NGS QC is with Linux. So if somebody is not having the Linux, the same tools are integrated with Galaxy server, one can use there itself. So now once we have our neat and clean data, filtered data, remove with the no presence of cut adapter sequences, then we have to go for the alignment. So to do so, first we have to index our genome and then align. So indexing of the genome, because as mentioned, if you are using the top hat, then we have to build the index using the bow tie tool and then use the alignment. So this is the alignment command. If somebody is using the top hat, this is the command one can use. It's very simple command and then the alignment will happen. So once we have done the alignment, after that there is a tool called coupling. This is the command for the coupling. This will quantify your transcripts and report the transcripts in FPKM or RPKM. And if any tool you are using, do not give RPKM, FPKM, but it will give you the raw read count. And I have shown you the formula for this. From this read count also, this can be calculated in an Excel very well. So now we have the raw read count from one sample. We have raw read count from the sample 2. Now we have raw read count from sample 3. Now in the next step, what we are doing, first we are merging all these 3 files together. And then from this third file, we are doing the differential. So we are doing 1 versus 2, 1 versus 3, or 2 versus 3. So we can do the differential in this way. So what does this mean? What are the common transcript? What is the unique transcript in each? And what is absent, not present? Like that we can compare between the samples. Using cuff merge to merge, and to do the differential like this, we use the cuff diff. So this is the command for cuff diff. Cuff compare. Cuff compare will compare. So this is an optional step. This will also give you, so you take this and then we can generate a visualization of this for heat maps or volcano, and can see what are the genes, how they are overexpressed or down expressed. We will see all that. So now, so this is just the whatever I have mentioned the tools. This is the description what they do. So these are the so these files to know, it is very important. So cuffdiff output files generate these many files. So FPKM tracking files, this calculates the FPKM for each transcript, primary transcript, genes in each sample, and it writes the different files. Primary transcript and gene FPKMs are computed by summing the FPKMs of transcript in each primary transcript group or gene group. There are 4 FPKM tracking files and their format is as. So it will give you the isoform file, which you can use to know the splice. If somebody would like to do the splice analysis, this file can be used. This is the gene raw data, gene count, read count. CDS, this is the transcriptome coding sequencing, whatever, because when you have messenger RNA, the entire messenger RNA will not code for your protein. So only the part of it will code. So that is your coding sequences. So it will give you a coding sequencing FPKM also, and then the TSS group FPKM file. So primary transcript FPKM. So if we have to do the primary transcript mRNA quantification, we have to use this file. Count tracking file, cuffdiff estimates the number of fragments that originate from each transcript, that is from the primary or the coding sequences. Primary transcript and gene counts are computed by summing the counts of transcript in each primary transcript group and gene group. Right? So these are the files which we get after cuffdiff tool, and then these files can be used for different purposes. So this gene file which is written over here, this can be used for down. So BAM file also, it's very important when we do the alignment, we get the BAM file. So this BAM file can be used for variant analysis. So someone is interested to know the variants in your transcripts. So this BAM file can be used, and there is a pipeline called GATK pipeline. One can use GATK pipeline for variant analysis. So now let us look for the differential expression. So I told that you can use cuffdiff for the to get the genes what are differentially expressed. So now how to do the analysis? So there is a tool called DESeq can be used. So DESeq 2, DESeq, AGR, Lima, these are the R packages to do the differential expression analysis. So are the counts we see in gene X in condition 1 consistent with those of gene X in the condition 2? So again there are sizing factors, estimate the library sampling depth, more stable measure for the coverage, mean ratio between conditions, variances. So this all it takes care. This tool takes care of all these factors. So we have map data, we have normalized expression data, we have read counts. So based on that, it calculates the differential expression for each sample. So what is the inference from this? Before we go and do the put our head into the analysis, just let us sum over here. So this is a very good paper. One should go through this paper. Gaining comprehensive biological insight into transcriptome by performing a broad spectrum RNA-seq analysis. This particular paper gives the insight about RNA variant calling, RNA editing, RNA fusion. The authors of this paper, they have examined both short and long reads RNA-seq technology using different tools and the combination. So they have analyzed total of 39 analysis tools, which gives the combination of 120 numbers and 490 analysis involving only 15 samples with a variety of germline, cancer and the stem cell dataset. So if somebody would like to know more depth in the RNA-seq analysis, how to design, how to do analysis, what different kind of analysis we can do, one should read this particular paper. So now let us, I'll stop sharing my screen over here and we will see some tools how we can use and how we can generate some visualization of our data. So I'll just stop sharing this screen. And we will just go and I'll give you one SRA number. Yeah. So I'm putting one number in the chat box. Okay, I'll show you also how you can go. So first let me share my screen again. So here, so what we have to do, go to Google and browse for ENA. So then this page will open and it will say the first link is European Nucleotide Archive. So click on that. Then you will see this page. And I put one number in the chat box. So take copy that number and put in this box, search box, and search. Then you can see this page. So you can see that there are two reads. Now go to Galaxy and use galaxy.eu. So you can create login over here, and when you come to this get data, paste and fetch data. So from here, click on this, so this fastq.gz. If you will put your cursor here and do the right click, it will say copy link. So copy this link and then paste over here in this box. Then go back to this screen and then again for the another read, copy the link address and then paste it over here and then say start. So when you say start, it will automatically download from the website and take your data over here. So now our data is here. So we can edit this name also so that our name will be little shorter. So we can just edit the name from here. So we can edit from here. So this is our sample one and save. Correct. So now in the pencil, if you will put, you can just edit the sample from here and then save. Now instead of that big name, we have a smaller name. So we can give another name also. Now we have this and then in this search tool, we can put fast QC. It will search for fast QC and then from here you can select your reads and then just execute. You do not need to change anything and just execute. Same for another file. So we have to do QC for both the files. File number 1 and file number 2. From this first drop down menu, you can choose both the files one by one and do the QC on that. Because to look at the QC before we proceed for the downstream analysis is important. So it will run and it will take some time to perform the QC. But to understand how it looks, we will wait for some time. Meanwhile, it gets finished. If any question is there, please do ask. So if you, this is Galaxy. If somebody would like to download the Fast QC, one can go to Google and write Fast QC. Fast QC for Windows. So when you go to www.barbaham.ac.uk, you click here and from here you can download. So you can download for Windows, Linux, you can download for Mac. So whatever the system it suits you use, you can download for it and just install like a simple exe file. So which I have already done. So I'll show you how it looks. So go to downloads, search here fast QC. So you will get, when you will unzip that folder, you will get the folder like this. And if you will run underscore fast QC, so the window will open. So window will open like this. You can go to file, you can open it. And then from here, you can choose wherever you have saved your file. So I think I have one file. I can show you how to use that. Yeah, I have one file. So just take this file and keep it over here. And then the analysis will start. You can see it is reading and it is performing. And how it will give you the results. So it will give different values. So it will give you some basic stats, sample name, what is the base call it perform, which platform the data has been generated, how many reads it has generated, whether any poor quality flag is there or not, what is your sequence length, what is the percent GC. So this is the basic statistics it will give. So the same thing what we are doing here, it will give you the same thing here also. So it will take for a while. When you are running on the web server, it takes more time. But when you are running on your local system or server, your own server, then it takes less time, because here you cannot change the codes or threads of your analysis, so it takes automatically. But when you run on your local system or your own server, you can change those settings. So if you look at the base quality, so the graph is divided into three zones. One is pink color zone, one is yellow color zone, and one is green color zone. If the reads falls in the green color, and I told you that the Q30, your data quality should be more than Q30. So optimally it should fall in the green zone. If yellow, if you cannot generate another data or rerun the sample, you can work for some objectives, but not always. But if your Q30 for Q quality score falls up to 20 or below 20, then your data is not good to go for further downstream data analysis. Then it will give per sequence quality score. See how it is. This is your average quality of your read. So if you will see, it is saying 36 is your average quality. So it means that for your data, the Q is 36 approximately. Per base sequence content, so this is all equal. So there is no bias in base calling. Per sequence GC content. So blue line represents if your data is clean, if it is coming from the single type of data or single organism not contaminated with any other data. So then your GC or your GC content should follow at one peak only. So that is which ideally is represented by a blue graph, and red graph is for your samples. So you can see there is some contamination over here, but again depending on your data type of data, because this is a microbe or pathogenic data, so you can see this kind of variation. Otherwise you should be able to see the follow-up along with the blue graph. Per n content, so you can see that in your data n's are not there, all each and every base has been called. Sequence length distribution. From the basic statistics, we have seen that our sequencing length is of approximately 150 base pair. So if we look at this graph, this is also showing, so this is the length, these are the number of reads. So you can see that almost all the reads will have the length of 150 base pair. Sequence duplication. So you can see that there is no duplication in the data. Overrepresented sequence, there is no overrepresented sequence. Adapter content. So you can see there is no contamination of adapters. If adapters are there, you can see little rise over here. But if adapters are there in significant amount, this will be like this. So I can show you how it will be. So then otherwise your graph will follow like this. So if you can see your graph is like this at the end, it means that this is the adapter contamination. So which is in this particular data that is not there. So like that we have to read the QC report. This is the initial QC report. If you see the adapters or any other things, then you have to remove those adapters using cut adapt, and then again you have to come back to this particular tool, whatever the filtered files are there, do the QC in that and visualize whether still adapters are there or not. If not, then we can proceed further for the downstream analysis. So because this is taking more time, but I will just show you how you can do the alignment. So what this data is, this is this SRA number. If we go back here, we can see what this data is. So we have to look at this data. So this data, this transcriptome data is coming from Dino radio. So Dino radio is nothing. This is the zebrafish. So for when we are taking any data, we have to see whether reference is available or not. And as we know that for zebrafish, the well annotated reference is available. So if you go to another site, just Google it, iGenome. So there are different sites, different web servers. from where you can download the reference genome. So you can go to iGenomes Illumina. So this is the site for it. So when you go here, here is the list of the organisms, and it has reference genome from Ensemble, NCBI, UCSC integrated over here. We can download the reference from here. So here you will get the reference genome and GTF file both together, either from Ensemble, NCBI or UCSC. You can independently go to Ensemble, NCBI or UCSC and download from there, or you can download from here also. So it will fetch the data from there itself. So this is your reference. So what is the beauty of this? All the references, whatever the available references are there, we don't need to download independently. All the references are here. This QC is not done, otherwise I would have shown you. So if you will write top hat here, I'll just brief you about that. So top hat also, they have different kind of versions. So what is our aim to here? The alignment. And I told you that we need a gapped read mapper. So we need gapped read mapper. So when we come here, so is this single end or paired end? So we know that we have uploaded 2 reads. So it is definitely a paired end read. From here we can select single end, paired end as individual data set, or paired end as collection. So this is not a collection. If we have so this is one sample. So if we have more samples, we can say that, okay, this is a paired end as collection. So then we can give, so if we choose paired end as collection, then it gives a different kind of situations. Then you can select. But this is not the case. We have as an individual data set. So from here, we can select the first read. So first always we have to select the forward read, and then we have to select the reverse read. We cannot do other way. So first we have to always select the forward and then the reverse. We cannot select first as a reverse read or the then forward read. So always we have to select first forward and then reverse. So first when we have done this, so I'm just putting all that as a default. Now comes to the reference. Use a built-in reference genome or own from your history. If you do not want to use from here, as I mentioned to you, you can download from there and upload over here as you have uploaded the data. But if you want to use the built-in genome over here, then I have selected using built-in genome. So you can use from history or from genome inbuilt. So I'm choosing built-in because I know that for zebrafish, well annotated reference is available. So now when I select go here, I can see zebrafish here down. So you just put here zebrafish. So now zebrafish is there. Okay. So again, you just see which one do you want to use, because there are 3 releases. So before using anything, we have to see which one we would like to use, whether from 2014, 2017 or 2010. So usually the latest one will be the updated one. I can choose this, and I'm choosing, there is no a specific group, so I am not specifying my groups because it is only one sample, and I can just execute it. So then it will generate the alignment file. So it will generate accepted hit as a BAM file. So once I have done this, so here I cannot do differential because I have only one sample. But what I can do here, I can see what transcripts are available. Say suppose I have given some treatment to my zebrafish. So that is my sample. I have created some mutation in my zebrafish, and I would like to see whether now my any transcript changes are there with respect to reference, or any variation is there. So here I can do 2 type of analysis. I can see the transcripts alternative splicing. I can do the transcript discovery. Any new transcript is formed or what is the quantity of it, that I can do, or I can do the variance analysis out of this data. So once I have my BAM file, I can do the QualiMap. So all that tools are here. If you see, if you write the QualiMap, so QualiMap BAM QC, if you will run on that accepted BAM, automatically detects actually. So what is the beauty? You don't need to select the files. Automatically, whatever the next step you would like to perform, it automatically detects the file from your history and performs the analysis. So once you have done all these things, I have done this and downloaded the file. I'll just show you what else we can do on this. So we can see, so I have my reference, I have my sample. I would like to see what is the difference between those transcripts. So I can create a heat map. So to do that, to do the heat map, I have used a package called NMF. So if somebody is familiar with the R, you can do very well in this. So for this NMF package, my file needs to be in CSV file. I can convert Excel in CSV, or I can keep CSV back to Excel. So when I will do the comparison, I will generate my files over from here. So this will be in a text form. So we know that text format can be converted into Excel or CSV very easily. Just convert them. I can show you the file format for that. So I need a CSV file to do that. So this is how I can do. So D is my sample and C is my control. So if I have two samples, I can run a heat map and see how they are different. So this is my sample, this is my control, and this is my sample. These are my FPKM values, normalized one. So here this is my ID. If I am doing, because this file is generated from the de novo when reference is not available, so it will give you ID like this. But when you do the annotation, it will change into gene. If you are generating this file from the reference based alignment, then you will already have a read name over here along with the gene. So that is the only difference. So this is how you can prepare this file. Once your file is prepared, you can save as a CSV file. So once it is saved, you can download the package called NMF. So once you have download and install NMF package here, then from here you can select the NMF package and then call for the library. I have already called the library. This is a CSV file. So to read the CSV file, we use the operator read.csv. Then our file name, header is true, row name is 1. So when we run this, I'll just clear my all these terminals. Okay, so now I have called for the package. Now I am calling my file, I am reading my file. So my data is a 50 observation and 2 variables. 2 variables is means one control, one sample. And I have taken a top 50. You can take the entire, whatever the gene list comes, whatever the transcript number comes, you can create a heat map from the all the genes, how they look, how they are different from each other. So that also can be done. So but here today I have taken only 50 observation, that is the top 50 observations. So I am calling a X. I have stored my data in X. I am calling X. So this is how my data is. So this is my script. This is how many observation. This is 50 observations. So I changed a 50 observations and then I'm just generating a heat map. So I have stored my heat map. It will go and store in the same directory where you have your files are generating. So my file name is plot1tif. I have saved, this is my file name, how my heat map will be stored. I can go back in the same folder and see plot1 has been generated now. So this is how the heat map will look. You will have the sample, you have the control. Here all the genes will be listed and you can see how they are different from control versus sample. So you can see these particular transcripts are upregulated in your control. They are downregulated in your control, and these are the upregulated in the samples. And these particular set of the genes which are upregulated in the control sample, and these are downregulated in the samples. So this is the color code, how you can specify which are upregulated and which are regulated. So this is minus, so these are downregulated. Which are 0, it means that they are not present in the samples, and these red color ones are upregulated. So this is how you can explain your heat maps. So now to see the another way of the so this is only the upregulated and downregulated. If you want to see other way of expression, then you can do the volcano plot as well. So for the volcano plots, you can use the package called enhanced volcano. So there is another library which you can download, which can be downloaded and installed over here. So this is called enhanced volcano. So this will create your volcano plots. So for volcano plots, the same file which you have prepared in an Excel, you can put in the text format. I will show you the file how it looks. So it looks like which file we have taken, D versus C. Yeah. So this is the same file, but this is the log to fold change and the p value, and then it is saved in the text format. So we need to understand the file formats. For each tool, for each package, there is a file format. So to create the heat maps, the file format is CSV, but to create the volcano plots, it takes a text file. So I have prepared a text file for this. So this is the script for enhanced volcano. First I am reading my table. Because this is a text table, so I am giving a header as a true. So when I will run this, so I am reading my table and I am storing in res. So you can give any object over here, whether res or anything. I can change this also. So closed. I'll just open it again. Sorry. Yeah. So and wherever our files are there, we have to set the directory. So to set the directory, we can go to session and we can go to set working directory and choose directory. And then we can choose the folder back and then wherever our files are there. So like that we can set the working directory. So now I am reading my table. I can store in any of the file. Say suppose now I can change this. I can give any name where I am storing my file. So I'll just read this. So now I have to change here also. Now my file name has changed. So if I will read here, so this is the file it is reading. So now this is the script. Now here everywhere I have to change the file name. Here also I have to change the file name. So like that, if I read this, it will generate a volcano plot for me. So when it is read, it is working, and then it will generate a volcano plot over here. So it will take for a while. So now you can zoom also to see a bigger picture of your plot. So like that. So this is, so red color is your upregulated log to fold change. Red is your downregulated. This is non-significant. Like that we can interpret this. So this is the log p value. This is the log to fold change on the x-axis, and log p is on the y-axis. So we will get from this volcano plot, we will know which genes are significantly expressed, highly expressed, or lowly expressed. Like that we can interpret with this volcano plots. So like that, there are different another kind of visualization tools. So you can use all these things together. So let us see where we are up to here. Yeah, QC is done. We can just see. We can just refresh so that our top head can start. Okay, whenever it will start. So QC is done. We can just have a look at this QC for this particular data. So if we go here, click on the eye icon, then we can see here. So this is the basic statistic of this data. So this is the name of the file, conventional base call. This is generated from Illumina. These many number of sequences are there. There is no poor quality flag. The sequence length is approximately 100 base pair and percent GC is 47. So quickly we'll just see few things. So here as you can see that all my quality score is good, falling in the range more than Q20, Q30. Base sequence quality. If I go down, I can say the base quality. So this is good. So we can see that it is on an average 36, 37. So if we go to the GC content, so per sequence content is also good. Everything is being called properly. Now you can feel that how this GC content is different from the earlier what we have seen. So here in the earlier data we can see that multiple peaks over here, but now you can see that this is only a single peak which follows along with the blue line. So it means that our data contamination is not there. It is a clean data coming from only one type of sample. So per base and content, if we look at this, so there are no ends in my reads. So this is also clean. Sequence length distribution, we have seen that it is a 100 base pair length read data. So we can see that it is again 101. Overrepresented sequences are not there. It is cleaned. Adapter content. So we can see that adapters are not there here. So we don't need to do any cut adapt on this data. This data is good to go for the downstream analysis. So already we have said for the alignment, but I don't think so you will be able to see alignment because alignment takes a long time, and it depends on how server is busy and how server is free. So but I will show you the further steps how you can do. So once the top hat is there, you can do qualimap BAM QC on that to look how the quality of your alignment. So paired and what is the percent alignment. If somebody would like to use star apart from the top hat, write star over here, and one can use this RNA star gapped mapper for RNA sequencing data. So if you choose this, again it will ask you, this is individual data set paired, and you can choose the reads from here and then just execute. So again use the built-in index. We are not going to load anything. Built-in model, and from here we can choose our zebrafish. So you can use anything 14, 17, and automatically it will take all these things. and then just execute it. If you want any other parameter in the BAM file to be right, then we can do all that. But usually run default parameters over here and then use it. So this is how it will look. So now it is started, but it will take, because it is a huge data, it will take long, at least 4-5 hours to finish top hat. Star will take little more time because star takes more computing resources than the top hat. So I'll stop here. Now the session is open for question and answer. Anybody has any doubt, you can ask.
For me, no. I think it's everything.
Sorry?
I don't have any doubts.
Anybody else? So I am hoping that you will be able to use whatever we have explained, how to look at the data, how to proceed further. So I am hoping that you people will be able to use this in your analysis with your samples.
Thank you very much.
So I'll stop here and happy learning. We have more sessions. You can visit BDG Lifesciences and explore more sessions. Yeah. Thank you. Good night. Bye-bye.